Uh, yes, I lead this program called BBRB, the Biorepositories and Biospecimen Research Branch. We were formerly known as OBBR. We have reorg at NCI as well, and now we're a branch. We're part of the Cancer Diagnosis Program at NCI. And our group was created in 2005 to address some specific concerns that are listed here. One related to reproducibility of research results. So we've got all of these biomarkers that we can't reproduce and develop into diagnostics. And how this really affects diagnostic tests and the pace at which these can get to patients. So this slide is really showing graphical representation of the problem. So these are cancer centers across the U.S. Somehow the way systems have grown up for research, we have ended up with a situation for biospecimens, research biospecimens, where we can have very different collection, processing, and storage procedures, really different degree and type of data annotation, and the IT systems in which that data lives. As you may know, the scope and type of patient consent can be very different across sites. The access policies for getting specimens for research can be not so transparent. MTAs can be quite different. And this has resulted in really quite a variety, and it's been our task at NCI to try to develop and foster the development of standards in this area. So we first developed a set of standards I'll talk about, the NCI best practices for biospecimen resources. We developed a research program to look in a very deliberate fashion at how, if we collect and process and store specimens in different ways, how does that affect molecular profiles? It's an area where our science in genomics and proteomics and metabolomics and so on is so far ahead of the relatively ancient technologies that we use to collect and preserve samples. I'll explain more about these other issues. So in terms of standards, three main activities: the best practices, a new project trying to develop evidence-based best practices, the first of which I think was published today. And we've also worked internationally to develop publication standards. So our best practices were first rolled out in 2007, and comments then were why are you doing this? And then once it was rolled out, why didn't you do this sooner? What took you so long? The idea was really to try to develop very basic standards. And I think sometimes when I talk about this, I realize how much misinformation there has been about this, because this is not telling people to collect specimens in a certain way or to collect Cadillac specimens, something that's really quite beautiful. This is about, get patient consent, write down that you got consent, keep a record of that, and all the way to have a battery backup on your freezer if you're using public funds to collect and store specimens. It's really a framework document, not a cookbook and not SOPs. So the idea was that this would be a baseline. And we have recently done a survey, and I'll mention a little about that. This best practices, as I mentioned, covers all of the issues on this slide. I've got a lot of material, so I won't talk through everything. but really quite the smorgasbord of activities we do when we do biobanking, and you can find this on our website. In 2011, we updated several areas, including management and operations, some information on custodianship and informed consent based on some workshops we had. This is an area that is just going to keep changing. Informed consent, not to mention things like return of research results. This is really changing also, as well as additional appendices. And our survey, we wanted to know, okay, so we did this first in 2007. What does this mean to anybody? We have a lot of reports. We know that this has been translated in languages across the world and cited many times, but we wanted a little more hard data. So we developed a survey, and when the government wants to do a survey, they have to go through a very onerous process. We can't just ask you questions. So we had to get approval from the Office of Management and Budget for this, which we did. And we distributed this quite widely and got about 400 responses from across the world, 75% in the US. And we think that the people that were responding are really involved in biobanking. As shown here on the right, you can see that many of these respondents reported that they really do day-to-day work with biospecimens. Overwhelmingly, people felt that this was a good resource. And this is sort of an interesting question because it's one thing to have a sort of a framework and to try to perform to that framework, but how do you actually, would you judge an organization that was using this framework better or worse? And it seemed that people would feel better about knowing that a framework was being followed. One of the great things that happened over the past couple of years is the NIH, of course, is not a regulatory organization. We're a research organization. And it was our hope that one of the groups that actually goes and regulates laboratories would take up these standards. And the College of American Pathologists has done that and has developed an accreditation program for biobanks, which is very exciting. We asked about which things were most important. We're asking some of this because we have reduced staff, just like lots of people in the world, and we have to figure out how we're going to do this into the future. Overwhelmingly, we hear that people want SOPs, people want SOPs, people want SOPs. And they want more information about informed consent and so on. One of the first things we're actually working on right now is legacy planning, and I'll mention that later because we're doing some big projects where we really need legacy planning. We looked and saw that we didn't have that much about that. And really, when you're first starting a biobank, that's when you should have a legacy plan. How are you going to take this biobank into the future, and how are you going to pay for it? We asked about what kinds of additional topics. Here you go, SOPs, quality assessment metrics. This is something that we are working on. Guidance on sustainable business models and cost recovery, and I'll say a few words about that. And incidental findings. This is, again, a huge issue. I think you'll have these slides, so you'd be able to review this later for more details if you would like. We got some comments related to medically underserved and vulnerable populations. This is really important. I know this group, CHI, has been involved in the NCCP program. And education comes up over and over as a theme, you know, for this whole biospecimen area and for for reaching patients and letting them know what biobanking is all about. Our BRISC guidelines is for biospecimen reporting for improved study quality. This was an international committee that met to develop guidelines for journals and for, we had FDA people on this as well, with the idea that if you do a study using human specimens, you should say something in your study about how you collected the specimens. You should say something about that can let people know, for example, how they should at least start to try to go about reproducing a biomarker result. It also can help reviewers find problems. There have been some highly publicized situations where biomarkers were discovered and then there was a lot of hoopla about it, and the controls were actually sometimes from a retrospective collection that was stored in a freezer, and all the cancer patients were collected prospectively under very different conditions. Not only does this cause, from a scientific perspective, differences that you might be able to detect, but also can cause significant bias that can really mess you up. So this actually has taken off better maybe than we had even hoped. We published this in 3 journals in 2011, and is now being cited by Nature as something to go look at. They're not saying you have to do this, but is part of the landscape, which we're happy about. Our biospecimen research program, the idea again is to develop scientific evidence that says if you collect specimens a certain way, how does that affect molecular integrity? And what kind of information can you get out of that? Well, you can get information about how should you optimally collect for a specific downstream analysis, but also when you collect a certain way, what caveats do you just need to accept if you really need to collect in that manner? We do this kind of research through our Biospecimen Research Network program, through some of our other programs going on. This BMS study was through our GTEx collections program. We incorporate research about ethical, legal, and social implications of biobanking into our other work to try to get real data about how patients are responding to requests for participating in research studies. And in the case of the GTEx project, which I'll describe later, the LC research relates to how do families of deceased persons feel about being asked to participate in a large genomic study. And it asks questions in an interview fashion after the fact, so after the donation has taken place and the person has either said yes or no, goes back and asks the family, do you remember anything about this? Do you remember what you were asked? Do you remember anything about how you felt about it? Because obviously we want to be able to do that part of this better, because none of, you can't do biobanking without people. We can't do research without people being interested and willing to participate. Some of the questions we address in our BRN program is how is biospecimen molecular integrity affected by specific pre-analytic factors? And an example is looking at ischemic time and time and fixative for FFPE tissues. Again, how should specimens be collected, processed, and stored for specific downstream analyses? We're doing some work, for example, on the effects of variation in blood collection, processing, and storage on proteomic analysis. Once the specimens are in storage, how do you know if they're okay to use? And this area of research and development really is developing molecular stability tools that one could use. This is a couple of slides that list out some of the projects that we're sponsored It's really too small to read, but these are 3 projects that relate all to blood and serum pre-analytic variation. And in one of these being performed by Caprion Proteomics, they are asking whether different blood collection, processing, and storage procedures result in significantly different proteomic profiles. And some of the results are shown here. And some of it's really good news because people who do proteomic studies really worry about how quickly do they have to spin the blood, and can they freeze thaw, and is that going to be a big problem or not? And they found that less than 24 hours at room temperature, they really couldn't see major differences. This is great. I mean, we want to find good news from these studies as well as where there are critical issues. It can't all be bad news. But 48 hours, yes, they did start to see degradation. If this heats up to 37 degrees, that's a problem. So if we are collecting in the jungles and we have no way to refrigerate the blood, then again, back to caveats, we have to accept the caveat that there's going to be degradation. And if you're going to go and spend millions of dollars on proteomics, you're going to have to keep this in mind and try to understand, have some kind of way of measuring that variability that you're introducing that you can't help. There's nothing you can do about it. And several other projects are listed here that relate to breast cancer tissues, and a large study looking at colon cancer and metastasis to the liver, ischemic times. All of these projects were contracts. We were, when our program first started, we were awarded funds for contracts. These were all competitively awarded RFPs. We also have a project going on that's looking specifically, and Rachna, who's in the audience here, is leading this for us. The Biospecimen Pre-Analytic Variables Program, and this is being done at multiple sites, and we're testing effects due to time and formalin, short times in formalin, and sort of over the weekend times in formalin to mimic what would actually happen if you had surgery on Friday. Because there are some reports that this can be problematic for molecular testing, and effects due to delay to fixation. We're planning a new experiment that will look at freezing because even though people are religious about how they feel about how they should freeze tissues and what temperature they should be stored at, there's just really not enough data to tell us the information we want. And we'll be doing a variety, we're doing some of this right now, of analysis really from multiple platforms to try to understand how, in this first set of experiments, how FFPE influences the results of some of these different technologies. We also have had an annual symposium, the BRN Symposium, which we're planning again for 2014. And the rules, does anybody remember reading about the GSA having meetings in Las Vegas? Anybody remember this? Yeah. So our rules for running conferences have dramatically changed, and we can no longer run a conference of the size that we used to, even though we did not hand out bicycles or even pens. So we're going to be doing this as a co-sponsored meeting, which I think is actually great because this was always well attended by industry, by academics. It was a pretty interdisciplinary meeting that in its last iteration had about 450 attendees. We have a literature database I'll mention something about, and a couple of economic surveys going on right now. Our literature database is known as the Biospecimen Research Database, or BRD. This has about 2,000 articles related to this whole area of biospecimen science. You can find it on our website, biospecimens.cancer.gov. And we have 3 part-time PhDs who work from home and read all of these papers and catalog them into a searchable database and summarize the findings. And there's actually very little about urine biospecimens in this database because there just isn't that much published. And we're actually doing meta-analyses of these as well. To do this, we had to develop a language for how do you catalog these kinds, how do you describe and catalog these kinds of studies. And so, you know, for example, we've called pre-acquisition factors as diagnosis, age, gender, and et cetera. Preservation, examples, clearing agent, embedding medium, time and fixative, and so on. Analyte extraction and purification, platform-specific methodology, so we catalog along these lines. And the language for this, we also had to develop in order to do our experiments, and there's just recently, the College of American Pathologists has published a call to standardize these kinds of data elements. So there's really a move to try to start recording this information, which is extremely difficult to start to do in the clinical setting. Nevertheless, we're glad that there's some attention to this. We have, as I mentioned, an economic survey going on right now. You can find this on our website as well. So financially sustainable biobanking, you can help. So you go to this site and you can, if you're willing, spend some of your time filling out this survey. The goal is to gain a greater understanding of the true costs associated with startup, maintenance, and long-term sustainability of biobanks. We have a second survey that will be coming out shortly if it is not already out. This survey is going to result in a published report. The second survey is actually going to populate a web-based tool where you can actually go and query the tool to get real cost information, and that's going to be based on actual survey data. So this is also filling a huge gap because there just isn't information about this. Regarding patient advocacy, we've heard this over and over, but this was a particularly notable comment from our survey, that patients need awareness of what is tissue, why is it important to research. A pilot study showed 61% of patients thought they were going to die when asked about donating tissue, confusing it with organ donation. Not that surprising when you think about it, but so we have a new patient brochure that is publicly available from the NIH Publications Office. And right now the new Spanish language version is in printing. And this is apparently for NIH publications, this is flying off the shelf, so we think that this is helpful. It's very comprehensive. This went through a couple of focus groups and was brought down to a 7th grade reading level. And it's actually it's a pamphlet, I should have brought some with me, but several pages long, but very simple. So feel free to use this. We also do quite a lot of outreach. I'm only going to mention one of the projects because one of these is taking up a heck of a lot of our time, the GTEx Biospecimen Collection and Management. This is a NIH Common Fund project that was started by Francis Collins when he was the director of the NHGRI, or the National Human Genome Research Institute, and before he became the director of the NIH. And it's sort of, I guess, what I would call a next generation human genome project. And this is looking at the normal genome. So the basic idea is that if we can discover through all of these GWAS projects that parts of our genome indicate increased susceptibility or resistance to disease, that you should start to see changes in those areas of genome before people develop disease. And so this is a big project trying to understand how genomic variation affects differential gene expression in normal tissues, and is already producing incredible data that's on dbGaP and is being analyzed all over the world. It's quite unique. But the challenge was to collect 20 or 30 different tissues in the pilot phase from 190 individual donors. And the catch is we needed high quality RNA. So even though for 20 or different tissues, we'd obviously be going to deceased individuals, we had to have high quality RNA. And so unbelievably, I still feel, because I helped get this off the ground, this met the pilot goals and now is in scale up to 900 individuals. And there's an access policy, actually, notably, for using the residual tissues from the project. This is a diagram of how this works. So for us, both this project and the BPV project, where we have ceased being people who just work on standards, and here we are collecting specimens every day. So the specimens are coming in through two main contracts from NDRI in Philadelphia and Roswell Park Cancer Institute. Each group has partnerships with organ procurement organizations. And under NDRI is the ELSI study I mentioned earlier about asking families, the families of the donors. There's a comprehensive biospecimen resource. There's a comprehensive data resource through separate set of contracts. So this is, contracts being led across the NIH. There is a data center at the Broad Institute in Cambridge. There's a brain bank in Miami that's receiving the brains, and so there's quite a traffic pattern through all this. And the National Center for Biological Information, NCBI, is putting out all of this data. There's also a grants program for developing fairly novel statistical methods for analyzing this data, because their goal, initial goal, was to identify expression quantitative trait loci, or EQTLs, across these different tissue types, across different individuals. So this keeps us pretty busy. And I wish I could say out of trouble, but it's not true. So we did some biospecimen science work in this project also. And we were looking specifically at whether the initial preservative that was used for the project, which is called PAX-gene tissue, we wanted to know how this stood up next to freezing in liquid nitrogen, which we would think would be the gold standard, and dry ice. And secondarily, to look at post-mortem interval and how that might affect RNA quality. And just, very brief results. This is, we're waiting for data to finish up the publication, the manuscript on this, to put it out for publication. The PAX-gene preservative, there's a number of publications on this already. I mean, this is excellent stuff. It gives much better morphology, as you can see, than nitrogen or dry ice, which is not too surprising. But in fact, the RNA integrity numbers are quite good from the PAX gene, and you can see that 4 hours, 6 hours, 15 hours, this is quite superior at more extended postmortem intervals. So just in the last couple of minutes, I want to mention this new project that we're doing, because there's quite a gap in terms of, you can develop standards. That's kind of as I mentioned, framework about how to run a biobank. We can do this somewhat exquisite, detailed research about what really happens to molecular integrity when you collect things in different ways. But how do you take that data and really try to change biobanking practice? How do you try to change clinical practice? It's not easy. So we're working on this concept of biospecimen evidence-based practices, which would ideally be state-of-the-science procedures that would incorporate evidence from the literature. So is there evidence out there? And talk about this with experts so that we don't invent something that no one can do that doesn't make any sense, and make these publicly available. The first one we tried was for freezing tissue, because we thought this would be really easy. I mean, you take a piece of tissue and you just put it on some dry ice and you're done, right? But we looked into the literature to try to see what evidence really is there. And the questions we're asking are, should ischemia time be minimized, and why? Because everyone says, well, try to get this into, preservative or ice as quick as possible, but do we have information that really tells us why? Should tissues be placed on ice or left at room temperature? Question of goopy OCT or no OCT. Does it really matter if you don't have nitrogen? Is dry ice okay? What about things like nitrogen or dry ice cooled isopentane as a freezing medium? What about shipment temperature, and so on? So we combined efforts really with our curation efforts from the literature database and the work coming out of our research projects to develop these documents complete with literature summaries. And this is the one that I believe is published today, and you can find this on our website as well. So this is about snap freezing of post-surgical tissue biospecimens. The idea is this is not an SOP, but an evidence-based guideline. And one page I'm showing a page from the procedural guidelines, and skipping some pages and going to the summaries of literature evidence. And you can see just briefly that in some cases there isn't much information. This is about incubating specimens on wet ice as opposed to room temperature. A couple of papers talk about this, but it's nothing you can really grab onto. Whereas there's a pile of papers talking about ischemic time. They don't always agree, but we try to make that clear. We're not trying to invent information where there isn't, but we're just trying to make it more transparent. And one of the places where I think that this kind of information I hope could be helpful is when a new research project or a new clinical trial is being launched and groups are meeting to talk about what should the SOPs be. And these meetings can just, this can go on for 12 months or 18 months, and a lot of it's opinion-based. And wouldn't it be great if there was something evidence-based that you could use to speed up that conversation, because then you'd be speeding up the clinical trial, and then you'd be speeding up getting a diagnostic or a drug to a patient. So in progress on this is something new for proteomic analysis, because we have a lot of information on this now, and we'll be using really a fit-for-purpose model. We asked about priorities for this in our survey. So what do people really need? What would you be interested in having us develop? And this is, a graph of the kind of information. We got a lot of People want to know about storage conditions, about handling. They want biospecimen specific information, etc. And we got lots of comments. So a few comments shown here. I think FFPE-related evidence is important because this material is usually collected and processed within the healthcare setting rather than in a research setting. So there's much variability, and we are trying to address that. And a comment here, all the topics are important, difficult to rank, and they're interested particularly in nitrogen for anything other than viable cells. Countries in Europe are skeptical to use liquid nitrogen because of environmental factors. There are countries in Pittsburgh that are also skeptical of this. I mean, this is just, you know, it can be hard to get nitrogen into a pathology laboratory for safety concerns. So wouldn't it be great to just have better information about whether, you know, whether that's really important? So in closing, I would just ask you to let us know. We are in the planning phases of our next ask. You know, what's our next program going to look like? And we really need input. So let us know what your challenges are and what problems you think really need to be addressed. In particular, related to diagnostics development. Our team is shown here. We've got a really dedicated group of people and a pretty hard task, and we're really grateful to all of them. And thank you so much for the invitation, the opportunity to talk with you today.